These are the notes for AP Biology, transcription and translation, and post-transcriptional modifications. So far in our notes, we learned the history of DNA, we learned the structure of DNA, and we learned how DNA replicates. So what is the point of DNA? And you should have learned that DNA stores hereditary information. And so in order to express that DNA, we need to get a protein. So DNA holds the code for a protein. However, DNA is in the nucleus. So we need to get that code into the cytoplasm so that we can make a protein. Because if you remember, ribosomes, which are in the cytoplasm, is where the proteins are made. So we have two procedures, transcription and translation. We're going to do transcription first, and then we'll get into translation. So transcription is a process in which the RNA is going to be made. So RNA is how we're going to get the code from DNA out of the nucleus. So we need to make an RNA. And the RNA that we need is going to be mRNA. The M stands for messenger, and it's needed to carry out the genetic information from the nucleus to the cytoplasm. So we're going to learn about RNA, the structure. As you can see from the diagram, RNA is single-stranded, where DNA, remember, is double-stranded. Another difference you can see right away is that RNA has uracil in it. There are no thymines in RNA. So adenine is going to equal uracil. The other difference is that we still have a 5-carbon sugar. We still have a phosphate group in the base, but our 5-carbon sugar is called ribose instead of deoxyribose. And all that means is that we have an extra OH group, a hydroxyl group, on the sugar compared to a deoxyribose. And the other difference is that there are 3 types of RNA, and all 3 types are required to make our protein. First type of RNA is mRNA, messenger RNA. This is the main RNA that we need to make our protein, because mRNA is going to get the information from DNA and bring it to our cytoplasm. It has the letters, and those sequences of these nitrogen bases are always going to be in groups of 3. So like ACU, UCA, whatever they are. But we call those 3 bases codons. Codons are always written from 5' to 3', so remember that rule. Each of these codons code for an amino acid. And if you remember, there were 20 amino acids. And so basically mRNA, its function is to deliver hereditary information to the cytoplasm for translation. The second type of RNA is tRNA. T stands for transfer. And basically it's going to take the codon and decode it, and specifically get that code for a particular amino acid. It contains an anticodon, which is the opposite or the complement to the codon. So if our codon read ACU, that's the codon, then our anticodon would be UGA. So that means complement, or the opposite letter, according to Chargaff's rules. What the tRNA is actually going to do is bond to RNA, mRNA, using these anticodons. So you definitely need to know that word, anticodon. The last type of RNA is rRNA, and that is ribosomal RNA. And so that is the RNA that is found inside the ribosome and is structurally responsible for actually making the protein. Alright, so once we make our RNA, we need to translate it to get our protein. And so that procedure is called translation. So translation is a process by which mRNA is read using a genetic code. So I will show you the genetic code and refresh your memory on how to read it and understand it. And what the genetic code is going to give us, it's going to code for a specific amino acid. And if you remember, amino acids are the building block to making a protein. The genetic code, I'll show you in a minute, is finished in 1964. And there are 64 total codons on there. And those all code for only 20 amino acids. Translation occurs in the cytoplasm and in the ribosome, not in the nucleus like transcription does. Translation requires all three forms of RNA, the mRNA, the rRNA, and the tRNA. Okay, here's a genetic code. It's similar to the one that's in your notes. And so if you don't remember how to use this, I'll explain it really quickly. You have your letters in sets of three. So let's say your letters were A, C, A. You're going to take the first letter and get that from the left side. So that determines what row you're on. And so for our case, actually, is A. We're on the third row. Our second letter is C, and so that's up at the top. So C means we are going to be in the second column. So that means we're in this box somewhere. And then A is going to refer to the third row of that row. So that's going to be THR, which is 309. Notice that if you look, there are 20 of these amino acids. And sometimes the genetic code does not have these cheat codes on there. So you actually need to be able to use first letter, second letter, and third letter. So because a lot of the work is going to be, meaning the protein being made is being done on a ribosome, we need to know what a structure of ribosome is. It's not just a small dot, that's found in the cytoplasm. It actually has two subunits, a large piece and a small piece. And the small one actually fits into the large one. So when protein synthesis is occurring, the two pieces of the ribosome are together. When protein synthesis is not occurring, they are apart. There are 4 parts or sites on a ribosome. You have tape, T-A-P-E. T site is where the tRNA is going to enter. A site is where the anticodon is going to bind to the codon of mRNA. I'll show you a picture of this in a minute. The P site is where the tRNA is going to move to after the A site, and then the E site is where the tRNA will exit. So these pictures are a little bit different than yours, but it's showing the same thing. So obviously this is your small unit and this is your large unit on the ribosome, and they actually fit together and form the ribosome that's going to be used for protein synthesis. And actually when they're oriented, it looks like they're actually backwards in that the small is on the bottom and the large is on the top. It doesn't matter. But you have the 4 sites. You have T, which isn't being shown on mine. But T is where the tRNA enters. So the tRNA enters in the ribosome over here, and then it bonds here, and then it moves to the P site, and then it exits out the E site. So as an overview of what we're doing, we're basically taking DNA, or the gene on DNA, and we're taking that gene and making the protein for it. So we start with DNA, we do transcription, and then the result of that is having an mRNA. And then we do translation, and after translation, we will end up with our protein. Another way to look at it is looking at the language. Transcription is basically taking DNA nucleic acid language and turning it into RNA nucleic acid language. All right, so let's go into the details for transcription. Obviously, their job is to make mRNA. So we have DNA. We're going to need to obviously unwind it. But what's going to happen is we're going to have a template. That is our transcribed DNA strand. And then we're going to have the other side of the DNA strand, which is going to be our coding strand. The untranscribed DNA strand, or the coding strand, is going to be the same sequence as the RNA. So there's going to be a transcription bubble involved, and we have an enzyme involved as well. It's called RNA polymerase. So as you see on the diagram, the coding strand is here. That is the one that is not going to be turned into RNA. Our template strand right here on DNA is the one that's going to be turned into mRNA. And then notice and write it down if it's not on your sheet that we are building RNA from 5' to 3' direction. And we will see that in the future slides as well. And so you can see that RNA is being made right here, and the complement letter is being built, and that is all being done by an enzyme called RNA polymerase. So there are actually 3 different types of RNA polymerases. These are enzymes. RNA polymerase 1 is only going to transcribe our RNA genes to make the ribosome. So that one's involved with making our RNA. RNA polymerase 2 is the one that's going to make mRNA, and RNA polymerase 3 is the enzyme that's going to make tRNA. Each of them have a specific promoter that it recognizes to get it all initiated, And obviously the one that is in red here is the main polymerase that we're talking about, because that is the one that is going to make our mRNA. So how this works is there is a binding site where the gene is. So when you have a gene, there's a TATA box, basically is what they call it, TATA letter sequence, and that is part of a promoter. So the promoter region is a binding site that occurs before a gene. And so the promoter region contains different sites. One thing is the TATA box, and then there's an enhancer region, which is a binding site for activators. Activators are just things that will activate the gene or turn the gene on. And then there's also a silence region, which is a binding site for repressors, and repressors would turn the genes off. So this is kind of how it all gets started. So this whole initiation complex also contains transcription factors, and those are the purple circles that you can kind of see on the diagram. And so they bind to that promoter region. And so basically transcription factors are just a bunch of proteins, and they're going to bind to the DNA, and they can turn transcription on and they can turn transcription off. But what happens is we have a trigger or a I can't think of the word I'm thinking of, that will bind RNA polymerase to DNA, a signal, and that allows RNA polymerase, which as you can see is the big green bubble on the bottom, to actually bind to DNA so that we can make our RNA. So DNA is on the bottom here on this picture, and so you see it says T, G, G, T, A, C, A, etc. And so what happens is RNA polymerase bonds to this DNA and actually starts making the RNA. And so it makes the complement letter to the DNA letters using Chargaff's rules. It matches RNA bases to the DNA bases on one side of the DNA strand. And remember, this is occurring from 5' to 3'. All right, another concept that we have to go over is that basically in a gene, there are pieces of it that do not code for the protein. So eukaryotic genes are not continuous. There are exons and introns involved in a gene. The exons are the real portion of the gene, and so those are the blue portions on my picture diagram. Those are the ones that are going to be expressing the gene, and they code the DNA. And then you have all the introns, which is the orange part, that do not code for the gene. They're just in there, and we need to actually get them out. So the introns are going to come out. Okay, so here is our DNA. Okay, and so then we do transcription, and now we have a RNA, and it is a primary RNA transcript. And you can see it contains exons and introns, so they call it pre-mRNA. So label that if it's not labeled on your diagram. So basically what happens is that pre-mRNA, or that early RNA that's made, needs work after transcription. It needs to be modified. It is the primary transcript. So what we're going to do is do mRNA splicing, okay? And basically we're going to take the introns out. So you can see all the orange pieces get cut out of the strand, and we're left with a mature RNA transcript that only contains the exons. And so now it is called a spliced mRNA strand. Obviously this is kind of a big deal. When they're splicing that RNA, they need to not make any mistakes. It needs to be accurate. A single base added or lost is going to throw off the whole reading frame. And so if you see the DNA, and here's your RNA, okay? And then if there's a mistake, you can see what happens here. We had a substitution that was supposed to be AUGCGGU, and instead it looks like we got an extra G in there. And so that, when we end up doing the translation, is going to change the protein. It changed the amino acid. And so it's massively important that the splicing is accurate. So how is the RNA spliced? It is done by enzymes. They're called SNRPs, and basically that means small nuclear RNA, but they are an enzyme. And so there are several of them doing this job, and you can see that they actually bond to the RNA. They recognize the site of the sequence that needs to be cut out, and they basically cut it. You can see they completely like remove it and then cut it and then paste the pieces of the RNA back together, and until the introns are completely taken out. So also, I just want to make sure I mentioned that when you have all of these SNRPs working together, that is called a spliceosome. All right, we're not done yet. We have a problem. mRNA has to leave the nucleus where it was made and go to the cytoplasm. It is very crazy that RNA cannot do that right away. There needs to be some processing to occur. The ends of the mRNA need to be protected because there are enzymes in the cytoplasm that will attack it and degrade it down. And so we need to add a 5' GTP cap. So basically, we're going to add this cap here, G3P cap. It's a chemically modified molecule of GTP, which is similar to ATP, and it's going to help the binding of the mRNA to the ribosome later, and it's going to protect the mRNA from being digested by these enzymes in the cytoplasm called ribonucleases that will break it down if that cap is not there. On the other end, we are going to add a whole bunch of A's, adenines. Actually, they add 100 to 300 adenines, and those are added to the 3' end of the mRNA. And so doing that will help the mRNA leave the nucleus and get into the cytoplasm and keep everything stable. All right, so now we are done and we have a complete mRNA strand. So now we're going to do translation. Translation is taking the language of the nucleic acid, which was the RNA nucleic acid language, and turning it to amino acid language. So remember, in order to do that, we're going to use the genetic code. It is the code for all life, so all living organisms use this code to determine their amino acids to make their proteins. It is one of the common origins of all life. It is redundant, which means there are several codons for each amino acid. You can see that. So like the example that we had earlier was 309. See, there's 4 different ways to get 309. Okay, so it is a third base wobble, which means that third base might determine the difference between one amino acid and another amino acid. On the genetic code, there is a start codon right here. Okay, it is AUG. It is not on the genetic code on all copies. It does not always say start, so you're going to need to memorize that AUG is the start codon. It always codes for methionine. On the genetic code, it would probably say MET. And then there are 3 stops up here. And you do not have to memorize those because you'll actually get stopped from the code. But they are UGA, UAA, and UAG. All right, before we get into the details for translation, we need to learn about tRNA. Remember, they're called transfer RNA structures. And so these RNAs actually look like a clover leaf. Or it kind of looks like a T. Okay? And so there is an anticodon on one end, or the clover leaf end they call it. And that portion of the tRNA is going to bond to mRNA. Remember, it contains an anticodon, and that anticodon is complement to the codon on the RNA. All right, so tRNAs contain the amino acid. And so that amino acid is bonded to the tRNA by an enzyme called aminoacyl tRNA synthetase. That bonding of the amino acid to, you can see it bonded right here, to the tRNA requires energy in the form of ATP. That bond is unstable, so it really wants to get rid of its amino acid at the ribosome. All right, so how this works is we have initiation, elongation, and then termination. These are the 3 main steps. During initiation, our mRNA is going to bond to the 2 parts of the ribosome. Okay, you can see that right there. And then our first tRNA, which is right here, is going to come in through the T site, and it's going to bond and go past the A site and actually bond eventually to the P site. And it's always going to start at AUG. I don't know if you can see that on there, but it says AUG. And so the anticodon on there says UAC. And then notice that there is an amino acid, MET, right here that's attached to the tRNA. All right, so then another tRNA comes in, so now we're doing elongation. A tRNA comes in and bonds to the next codon. All right, so we have bonding. Notice that we have two amino acids next to each other. We're going to form a peptide bond between those two. And then this first tRNA is going to leave, which it does through the E site. And then we have a shift, and the next tRNA can come in. And it's going to continue this way, and so you can see that the protein is getting longer and longer. So eventually we're going to reach a stop codon. When the stop codon is reached, all the tRNAs release, the protein releases, and now we have a polypeptide or a protein. The protein that is made will have some type of signal on it, which is like an address label. It's basically telling the protein that this is your job and this is what you're going to do. And so all these different, there's all kinds of destinations that this protein can go to. It can be secreted out of the cell. It might go to the nucleus. It might go to the mitochondria, the chloroplast, like all these different destinations in the cell. So basically, this is a whole story, right? Starting with DNA, going through and making the RNA through RNA polymerase, going through and cutting out the introns, getting all the exons in after we had our pre-mRNA. So then we end up with a mature mRNA. We need to protect it by doing a GTP cap on the 5' end and a poly A tail on the 3' end. We're going to go to the ribosome and attach there. We're going to get tRNAs coming in and bonding to our codons with their anticodon with the amino acids, and then the next tRNA comes, and then we end up making this polypeptide, and once we reach the stop codon, we will release and we have a protein made. So this last part is on protein synthesis in a prokaryote. As you can see, they have one chromosome. They can do transcription and get the mRNA and then make a protein, just like we can in a eukaryote. But remember, there is no nucleus. Here's a difference on prokaryotes versus eukaryote genes. Prokaryotes, their DNA is in the cytoplasm. Remember, they have one circular chromosome. So that basically, and there's no nucleus, so there is their DNA, they call it as naked DNA because there's nothing really around it protecting it. And prokaryotes don't have any introns to worry about. They don't have to do any splicing. Where eukaryotes, our DNA is in a nucleus. They're linear chromosomes. The DNA is wound again on histones. It's a lot more complex. And they have introns and exons. And the introns have to come out for a eukaryote, where a prokaryote doesn't have to do that. So for prokaryotes, there is no mRNA editing. The ribosomes will just read the mRNA as it's being transcribed. So mainly the difference for translation for prokaryotes versus eukaryotes is time. It's obviously super fast for a prokaryote compared to a eukaryote. It takes a eukaryote around an hour, and a prokaryote is a lot faster than that. And then the other difference is that the prokaryote cell does not have to do any mRNA processing.